where he's been since 2001, appointed to the academic staff in 2010 and a full professor from 2015. As I hope you can see from the title, Ian's research focuses on signals between roots and shoots and mechanisms of communication between roots and shoots. He's published over 100 manuscripts in the area, and he's also an editor on Plant and Soil in the Journal of Experimental Botany. So without further ado, I'll hand over to you, Ian.
Brilliant. Thanks a lot, Rob. As many of you will be aware, Rob is also on the staff of Journal of Experimental Botany as well. And so it's actually great to have a conversation with him rather than just an email from him assigning manuscripts to me. So great to see you, Rob. Great to see you as well, Tim. And I know there's probably many on the call that I've met over the years, and thank you for your associations. So hopefully I'm sharing my screen now. Is that true, Rob?
Yeah, that's great. Thanks, Ian.
Brilliant. Okay, so it's nice to be in the room, as it were, giving a domestic presentation. I've seemed to have spent the last 12 months or so giving presentations in various parts of the world, mostly from the comfort of my living room. So it's good to be back in the office and giving a presentation to a domestic audience. Although up to the results of the local council elections last week, how long that will be a domestic audience and how long that might be an international audience, I don't really want to speculate on. But I've got some images here of me doing some experimental work, a little bit out of date now. Particularly over the last couple of years, it's been a challenging time to do experimental work. And as I've gone through my career, I've tended to manage researchers, and a lot of them have been working on a subject that's really dear to my heart, and that is long distance communication in the plant. And so that's the title of my presentation today. And as I've gone through my career, I've done less and less experimental work in controlled environment situations and gone more into the field, as you can see from some of these images here. So I'll give an outline of the talk. So it's divided into three parts. First, I want to consider root to shoot signaling and how that might affect shoot physiology, and then sort of flip it on its head, if you like, and look at shoot to root signaling and how that might affect root physiology. And if you're very interested in this topic, at the beginning of the week, some of my colleagues and I published a special issue paper in Physiology Plantarum. Don't go to it just yet. I think Physiology was that anxious to get it published that they put in one of the revised versions rather than the officially accepted version. But do take a look at that, because it will summarize not just ABA signaling, but also jasminates and strigolactones, which I've excluded from the talk today. The third part of the talk is sort of my foray into how this work on long distance signaling in plants might be applied commercially. And it uses some slides from a webinar I gave during the pandemic. And you can look at the website there if you're really interested in this topic. And that's the irrigation regime known as partial root zone drying. Now, it's quite sunny outside here today, so talking about water scarcity is not such a challenge for me in Lancaster today, unlike the normal situation. But I'm going to take you to Cambridge in 2018, and that was a very dry summer in the UK. Even in Lancaster, I saw my lawn wither and die in places which I've never seen in 20 years of living in Lancaster. And I had a PhD student who was working at Cambridge University Farm at the time with the group there, and she was doing some work on potato, which I think is a crop that a number of you in the audience will be very familiar with. And so my student was imposing different irrigation regimes with the team down at Cambridge. And when I visited her, she was really concerned that they were actually going to run out of water from this reservoir, and it was going to compromise her experimental design. Fortunately, this didn't happen, but it certainly demonstrated to all of us just how important irrigation was for this particular crop in the south of England. So these are some of the yield data that Katharina collected in conjunction with the group down there. And she had a classic 2x2 factorial experiment where she grew her potatoes in soils that were either compacted or not compacted, and either well watered or deficit irrigated. And you can see that, unsurprisingly, she got the highest yield with her plants grown under optimal conditions. And then with any stress, be it deficit irrigation or soil compaction, or indeed the combination of the two, yields were decreased. Now, Katharina also did a lot of measurements of the physiology of the plants over the course of the season. So she measured leaf gas exchange, she measured ABA concentrations in the leaves, and she also measured leaf water potential. And something that surprised both of us was the fact that even though the plants were clearly suffering with the various stresses, particularly the deficit irrigation, it was very difficult to demonstrate that these treatments actually had perturbed leaf water potential. Now, field experiments can be challenging for all sorts of reasons, not least the variability you get within the field. And so consequently, she did some experiments at Lancaster in controlled environment rooms. And this study that she did was just with withholding water from the plants. And you can see, unsurprisingly enough, when you withhold water from the plants, you exert a bonsai effect on them, and ultimately they wilt. Now, she also did some physiological measurements in her experiment, measuring stomatal conductance and leaf water potential. And very early after withholding water, you see that the stomata start to close. It's only much later in the experiment that the leaf water potential of these potato plants decreases. And it's only after 7 days you start to see wilting of the lower leaves of these plants. And so if you're using visual indicators of plant stress for irrigation scheduling, you've probably already substantially decreased carbon gain by these plants. So Katharina's not been the first person to do such an experiment. These experiments have been done with many species in many situations in the field in controlled environments. And it shows that in many situations, you can get appreciable stomatal closure and also growth limitation before you actually start to be able to detect changes in leaf-water relations. And it's observations of this kind from the 1980s onwards that led to the concept of root-to-shoot signaling, that the plant roots are detecting that the soil is drying, and then they are sending chemical signals to the shoot to actually regulate the stomatal physiology and constrain the leaf growth. And a number of researchers around the world elucidated the theory of these chemical signals, and that included some work that Bill Davis and Chanbai Zhang were doing at Lancaster not long before I joined the laboratory here. But there was a seminal review published by Michael Jackson, no, not that Michael Jackson, in 1993, where he articulated very clearly what was required for a compound to be regarded as a root-to-shoot signal. And that was that it needed to be generated in the roots, transmitted or propagated in the xylem, and that needed to have a physiological response in the shoot. And these graphs here just indicate some of the data collected very early in this idea of root-to-shoot signaling from different elements of these criteria. So we can see here, for example, that ABA can accumulate in the root system. In this case, the experiment was with detached roots that were allowed to dry. We can see that the ABA concentration in the xylem sap of the plant is very well correlated with the root ABA concentration. And then we can see, independently of whether the plants are exposed to drying soil or synthetic ABA solutions that are fed via the transpiration stream, the stomata close. So ABA seems to fulfill all the criteria of a root-to-shoot signal as defined by Jackson's paper. And so consequently, a lot of us, me included, spend a huge amount of time collecting xylem sap from plants in different ways. And you can see different ways of doing this via the cartoons here and also the images, and actually quantifying the xylem sap composition of plants exposed to drying soil. There is, of course, another way in which you can evaluate whether root-to-shoot signaling is actually occurring in planter, and that uses plant genetics and grafting, which is a surgical technique that has been known to horticulturalists for thousands of years. And some very interesting papers came out, in some cases 20 years ago, where wild-type tomato plants were grafted onto ABA deficient rootstocks. And similarly, for those who really wanted a surgical challenge, they managed to do this in Arabidopsis as well. And the sort of conclusion from these studies with reciprocally grafted wild-type and ABA deficient plants was that actually you didn't need root ABA synthesis to induce stomatal closure in plants exposed to drying soil. Now, I'm going to apologize in advance for the next slide because it's quite busy. What I've done is a sort of meta-analysis of all of these reciprocal grafting studies. I'm going to direct you to the right of the graph at the moment. And so here we have our wild-type shoots grafted onto wild-type rootstocks or ABA deficient rootstocks. And each of these studies here represents a report in the literature. And when you look at the consensus response for a wild-type plant, there's very limited impact of the ABA deficient rootstock, which fits the paradigm that the roots are, probably not that important in contributing to stomatal closure in response to drying soil. And the ABA data, effectively backs this up. It's interesting, though, that when you have an ABA deficient scion, you can substantially revert the stomatal phenotype, but also the ABA status of the plant, of the shoot, by grafting onto a wild-type rootstock. And so when you graft these ABA deficient scions onto wild-type rootstocks, you can demonstrate more effective stomatal control, and this seems to be ABA mediated. And indeed, it's interesting that the ABA status of the scion seems to determine the response to a root supply of ABA. And one particularly good recent example of this is a paper that came out last year that I did with my Spanish colleagues with whom I've collaborated with for a number of years. And in this case, again, we used wild-type rootstocks and then, wild-type scions. In this case, it was a commercial variety known as sugar drop tomato, and we grafted them onto either a wild-type rootstock or to rootstocks that actually overproduced ABA. So in this case, additional root-supplied ABA, when the plants were grown in slightly saline conditions, actually enhanced both the growth but also the fruiting of these crops. Now, whether that was a direct impact of ABA or whether that was an impact of additional root-supplied ABA on more effectively regulating the water relations of these plants, I think it's a bit difficult to actually make that distinction at this point. But clearly, rootstocks can exercise a dramatic effect on the physiology and also the commercial production of scions, particularly in crops like tomato. And another example here that we published even longer ago, looking at the impact of root-supplied cytokinins on the growth and development of the tomato crop again. And you can see a very similar effect with additional cytokinins supplied from the roots, actually enhancing the yield of these crops when they were grown with salinity. So I think it's probably premature to discard the concept of root-to-shoot signaling, even in the case of root-to-shoot ABA signaling. And certainly I think there's abundant evidence from other hormones that root-to-shoot hormone signaling can be very important in regulating the physiological responses of the shoot. And I'll direct you to that review article that came out at the beginning of the week that explores this concept with a little bit more detail. Now what I'd like to do is consider shoot-to-root signaling. And indeed, a couple of papers came out about 5 to 7 years ago, actually showing that the root ABA concentration is regulated by the shoot of the plant. And I just ask you to cast your mind back to one of the earlier slides showing that roots could accumulate ABA even if they were not physically attached to the shoot. So some detached root experiments satisfied the criteria that roots could synthesize ABA. Anyway, these papers came out that questioned the paradigm of root-shoot signaling and clearly established that the shoots were important in regulating the ABA status of the roots. And this experiment here was a girdling experiment, and this experiment was a grafting experiment. And I've got the results from similar experiments as I go through this section on shoot-to-root signaling of ABA and how that might affect root function. So as I mentioned, girdling is a surgical technique. I'm big on surgery, so I've talked about reciprocal grafting as a surgical technique. Here's a surgical technique known as girdling, where you actually remove the phloem from the plant. In this case, these are soybean plants. Big thank you to my colleague and former PhD student, Pedro, who was very skilled as a surgeon. Funnily enough, his father was a surgeon, and certainly my own surgical skills with girdling are a little bit deficient. I seem to be too keen in cutting into the plant, and a lot of my girdling attempts have not been as successful as Pedro's. What I would like to highlight is the amount of work that Pedro did here to measure the accumulation of ABA in the roots following his girdling treatment. And he made measurements 1, 3, and 5 days after girdling, and the impact of girdling was very much time dependent. So immediately within a day of girdling, there's a substantial decrease in root ABA concentration in the girdle plants, which subscribed to the paradigm that the shoots were actually controlling root ABA status. If we look 3 days after girdling, once the soil has started to dry in the latter 2 columns, we can see for the intact plants substantial root ABA accumulation, but then also for the girdle plants, they were still able to accumulate some ABA in the roots, as you can see here. It's interesting that after 5 days, we still have this considerable ABA accumulation in the intact plants, and there is still the same level of ABA actually in the girdle plants when they're exposed to drying soil. But clearly girdling is stressful for the plant. You're depleting the roots of carbohydrates supplied from the shoot. And so we can actually see for our well-watered girdle plants, they've substantially accumulated ABA. And so consequently, it gives the appearance of no root ABA accumulation in response to soil drying in the girdle plants. Now, so I would argue that the interpretation of such girdling experiments depends very much on the timing of your measurements in relation to the timing of the soil drying that's actually being applied. Probably more interesting than the source of the ABA within the roots, because I believe that it can result from both import from the shoots and also local synthesis, is the function of the ABA within these roots. And using these ABA overproducing tomato genotypes that Andrew Thompson created, you can see here a very clear effect that accumulating more ABA within the roots actually enhances the root hydraulic conductivity. Now, this may be very beneficial when the plants are grown in drying soil, because it allows them to continue to try and maintain their water status. But if the plants are grown under optimal conditions, we end up with this symptom here of water soaking, where essentially the root hydraulic conductance is so great that it's greater than it's better in providing water to the shoots, and the shoots are actually losing water to the atmosphere. And so, you get guttating tomato plants if they overexpress the biosynthesis genes for ABA in the root system of the plant. Another important consideration for ABA accumulation in the roots that it might be important in actually maintaining root growth in drying soil. And this is the work of Bob Sharp's lab. Bob was also a PhD student with Bill Davis here at Lancaster. And this is one of the papers from his PhD, where he grew maize plants in very long soil columns and simply harvested plants at different stages during a soil drying cycle. And what's quite interesting is that you can see that the root growth at the top of the figure was substantially increased very early in the drying cycle. And this occurred prior to any substantial leaf growth inhibition. And so Bob's lab has gone on to try and understand the role of root ABA accumulation in regulating root growth. And this image shows the responses very nicely. So these plants are all grown at very low water status. And you can see looking on the left here, in the absence of fluoridone, which is an ABA biosynthesis inhibitor, you get substantial ABA accumulation and substantial root growth. If you add fluoridone, the ABA biosynthesis inhibitor, you substantially restrict ABA accumulation and you substantially restrict root growth. What was particularly interesting about this paper from Bob's lab was it seems as though the role of ABA is to restrict the synthesis and action of the plant hormone ethylene, because if you added ethylene biosynthesis or ethylene sensitivity inhibitors, you can restore the root growth, even though you're not actually restoring the ABA concentration of the roots. What's interesting about ABA is that often its impacts are highly dependent on the water status of the plant tissue. And this is again an image here from looking at root elongation rate in these maize seedlings exposed to drying soil. And in this case, instead of using a chemical biosynthesis inhibitor for ABA, Bob's group has used these viviparous mutants that have low ABA concentrations. Now, from a commercial perspective, this is not ideal because they actually germinate while they're still attached to the plant. But they have been used within these experiments looking at root growth. And you can see that when the plants are in a well-watered state, additional ABA accumulation inhibits the growth of the roots. However, when they're in dry soil, you actually get the opposite response, that you get greater root growth in water-stressed plants if they have ABA accumulation. And the impact of ABA in terms of its sensitivity does seem to depend on how the water relations of the plants are placed. But to go back to the reciprocal grafting experiment, ABA also seems to have a role in terms of regulating numbers of roots. And we can see here that for the wild type root system of both tomato and pea, we have a limited number of adventitious roots. That can be reverted if you have an ABA deficient scion grafted to your wild type rootstock. And likewise with the ABA deficient rootstocks that have prolific adventitious rooting, you can revert this by grafting a wild type scion onto those ABA deficient rootstocks. And again, we have the situation looking at the root ABA concentrations, where the rootstock ABA concentration is largely determined by the actual scion. And particularly look at the reciprocal grafts here, where we have a wild type rootstock grafted on an ABA deficient scion with low root ABA concentrations, and likewise an ABA deficient scion grafted onto, ABA deficient rootstock grafted onto a wild type scion that has high root ABA concentrations. Again, I would stress that the value of the meta-analysis that I showed you earlier and showing that this particular example is a very absolute case of scion control of root ABA status. If you look more comprehensively across the entire literature, you can certainly see examples of such a phenomenon, but actually in some cases the root has more autonomy over its root ABA concentration. More recently, I've been doing some work with a Chinese group on rhizosheath development in the soil. Rhizosheath is the soil that adheres to the root system upon excavation. And I'm sure you've all heard about this because Tim and Laurie have worked on this at Dundee for a number of years. So I was able to supply some ABA deficient mutants to the group in Fuzhou in China, and they demonstrate an impact that in the absence of ABA, you actually get less rhizosheath development in both barley, but also tomato as well. And you know, it's interesting to know what the mechanisms that might cause this reduced rhizosheath development might be. So we looked at root hair length and also root hair density. So within the barley, it seemed to be that the ABA deficient plants had shorter root hairs, and consequently this seemed to have an impact on reducing their ability to bind soil in the rhizosheath. Within the tomatoes, the impact of ABA deficiency on root hair length was not that strong, but we did seem to have, particularly in drying soil, lower root hair density with ABA deficient roots. So, you know, this work we've done with rhizosheath development is potentially very interesting for a number of reasons. And I think the jury is still a little bit out on how important the rhizosheath is in ameliorating the plant responses to drought stress. A very nice paper from Tim's group last year on this, looking at some field experiments done at Dundee. So I've been collaborating with John Quinton here at Lancaster, and we're very interested in the ability of root hairs to potentially prevent soil erosion. And there does seem to be some emerging evidence that root hairs, by binding the soil, can mitigate the impacts of rainfall on erosion of the soil. Some recent work from one of my PhD students here suggests that at least in young plants, the rhizosheath can contain just as much carbon as the actual roots. But whether this is typical of more mature plants, I think we need to do some further experiments. The other reason why the rhizosheath could be very important is because it provides a niche for soil microbes, and this could be very important in terms of nutrient cycling. So to sort of conclude this part of the talk on shoot to root ABA signaling, I think it's only right that I consider I think what is a particularly rare case of soil microbes that can actually metabolize ABA. And so this is some work from one of my colleagues in Russia, Andrei Belemov, where he has isolated ABA metabolizing rhizobacteria. And we can see here from the graphs that the ABA metabolizing rhizobacteria have an impact in reducing the ABA status of the roots, but also affecting both root elongation and root proliferation. Of course, these bacteria are actually metabolizing ABA that is actually exuded by the roots. And it's interesting with Emma Sayer, who's a microbial ecologist here. I've been doing some work looking at the respiratory responses of soil to ABA addition. And it seems as though the soil will have a respiratory burst in response to ABA supply to it, even if the concentrations are probably much higher than would be expected to be in the soil as a result of plant action. But it's interesting that there seems to be some drought adaptation going on, that the ABA addition to soil that has been exposed to a long-term drought had less of an impact in terms of stimulating microbial respiration. So at this point of the talk, hopefully I've provided some concepts that ABA can be synthesized in the roots. It can travel up where it may have an impact on the shoots. It can also travel down where it can have an impact on root function, but also on the microbial complement of the roots around the root system. In the last part of the talk, I'd like to think whether we can use this information for commercial benefit. And there's been a number of studies increasingly over the last few years that have actually looked at transforming plants with overexpression of the NCED gene that is the rate limiting step in ABA biosynthesis. And I've provided a couple of examples of such papers here. So in this case with rice, even though the paper does overexpress the NCED gene in rice, they only reported the impacts of ABA deficiency on rice production. And we can see here that in the presence of water stress, with less ABA in the plant, you get less grain set. And it's unfortunate the ABA overexpression lines weren't profiled for seed set rate. Interestingly enough, under well-watered conditions, no real impact of the ABA deficiency on grain set. So I think it would be pretty obvious that you wouldn't grow ABA deficient plants in the field because it's difficult to produce seed from them, particularly if there's water stress within the environment. So I think there is some interest then in overexpressing ABA biosynthesis genes and see whether this can make the plants more stress tolerant. And this one example that I pulled from the literature seems to suggest that actually overexpressing ABA has a negative impact on seed weight, but also on grain yield when plants were waterlogged. And I think there's probably also similar data coming forth looking at responses to drying soil. And we need to think, well, why might this be? And these are a couple of papers that I found, highly influential over the years in terms of helping me understand this relationship between water use efficiency at a leaf level and water use efficiency at a field level. And so if we look at this study here, looking at the relationship between yield and evapotranspiration, we essentially get a linear relationship between the two. And so if we're in a water limited environment and we can maximize our water use, we should enhance the yield of the crop. The other way of doing this, of course, is to increase water limited yield at the same evapotranspiration. And under these circumstances, you'd argue that it's probably not going to be that beneficial to overexpress ABA within the plant because it could constrain your water use and therefore constrain your yield. Where overexpressing ABA may be more useful is in irrigated situations, particularly to reduce this idea of luxury transpiration. And this sort of idea of luxury transpiration has been sort of generated predominantly from leaf level measurements. And we can see here, looking at Andrew Thompson's ABA overproducing tomato plants again, the relationship between photosynthesis and stomatal conductance. And we can see here that across most of this relationship, prior to this inflection point here, actually the stomata continue to open for very little photosynthetic gain. So they continue to transpire. Now, you can see here, looking at the ABA overproducing tomatoes here, a substantial decrease in photosynthesis. There's one line that is a weaker overexpressor that falls around this inflection point. And so possibly that may provide some benefits in terms of reducing transpiration, but without actually suppressing photosynthesis and thus biomass production of the crop. So while there are genetic ways we could improve water use efficiency, there are also management ways we could do this. And that's what I've predominantly been interested in when I returned to Lancaster at the beginning of the century. It seems like a long time ago here now. So I've been focused very much on applying different forms of deficit irrigation. So by deficit irrigation, I mean giving the plant less water than it is been calculated that it would use. And in doing this, you can potentially save some water, and the hope is that you do this in a way that doesn't compromise the yield of the plant. And I've worked a lot on an irrigation technique known as partial root zone drying. And the idea behind partial root zone drying emanated from a laboratory experiment with split roots. So I'm certainly aware that one of Bill Davis's PhD students in the 1980s did some work with split root maize plants. I was talking to Manfred Stoll about this, and he very kindly sent me this cartoon from one of Julius von Sachs's drawings from the 1870s, suggesting a split root experiment in maize. Now, I don't know what the hypothesis of this experiment was. My command of German is not that great, but all I can say, given the complexity of this split root experiment, I'm glad I wasn't the student that was actually doing this particular experiment. It looks particularly challenging. The experiment that Peter Blackman did here at Lancaster was much more simple than that. The roots of maize plants were divided into two compartments, and then two different irrigation regimes were imposed. So either both sides of the pot were watered, or one side was watered and the other side was allowed to dry the soil. And the aim of this split root experiment was to try and break the link between soil drying and leaf water deficit. The experiment was done at a time when sort of leaf water status was accepted to control stomatal conductance and to control leaf growth, and various researchers around the world were just thinking of ways in which they could demonstrate that perhaps the roots were also important in mediating plant responses to soil water deficit. So with this experiment, a whole lot of measurements made of leaf water relations, leaf water potential, solute potential. And what's interesting is that actually the plants that were exposed to drying soil had slightly higher turgor as the soil was allowed to dry. And that seemed to be because the stomata were partially closing in response to drying part of the soil. So stomatal closure, presumably in response to root to shoot signals, was actually regulating the leaf water status of the leaves. So this is a nice piece of fundamental science. The sort of next step in the narrative was actually seeing what impacts this could have if you imposed partial root zone drying in a commercial setting. And so this is one of the early experiments done by Brian Loveys in Australia, where grapevines were grown with their roots initially split by plastic. Later on, they found that it was possible to impose discrete wetting and drying parts of the root system just using drip irrigation. And you can see here, looking at the results, that the partial root zone drying plants, or PRD plants, they had less water applied to them, yet they maintained the same yield of grapes. And this was possible because the grapevines were less vigorous, their growth had been restricted, and also the stomata had partially closed in response to this partial root zone drying. Now, this technique was widely disseminated to Australian grape growers and reported around the world, and certainly generated a substantial amount of controversy, I would say. Certainly, I've looked at a blog post from an irrigation symposium at around this time and seen some statements such as PRD does not offer any benefit over conventional irrigation, and there is nothing magic about alternating the wet and the dry sides. But the wet and the dry sides alternation is an interesting piece of physiology. Initially, when you withhold water from the dry side, the stomata start to close. But later on, if you continue to withhold water from that dry side, actually the stomata start to reopen again. And I think I understand the basis for that. So these two statements that I saw at the beginning of the century, when I returned to Lancaster to do some work on partial root zone drying, really got me thinking. And There'd been quite a number of studies comparing partial root zone drying versus deficit irrigation. And so I summarized a lot of these in a meta-analysis. So each one of these bars on the graph represents a separate study reported in the literature. And what I've done here is I've compared the yield response of partial root zone drying plants to the yield of deficit irrigated plants that receive the same irrigation volume. Now, if the technique itself is having no difference from deficit irrigation, so the technique is solely based on withholding water, you would expect all the bars to lie on this line of 1, which is the yield ratio of partial root zone drying to deficit irrigation. And there are some studies where there's no impact of the irrigation placement on the yield of the plants. But more often than not, it seems as though the partial root zone drying plants significantly out-yielded the deficit irrigated plants. And this got me thinking as to why this might be the case, whether the signaling from the roots of the partial root zone drying plants was different from the deficit irrigated plants. So I found a way of actually measuring the signals coming out of roots of split root plants. And I used a grafting technique to do this. This is my so-called two-legged sunflower plant. And what they allowed me to do was collect xylem sap from the 2 different parts of the root system, but also measure the sap flow emanating from both parts of the root system using sap flow gauges. And having collected the sap, I was able to measure ABA concentrations in it from the 2 different parts of the root system and from the leaf. And what was interesting was that according to the irrigation placement, I got very different responses of ABA concentration to either leaf water potential or soil drying. And what was very interesting was that as the soil started to dry, I got an elevation of ABA concentration in the plants exposed to partial root zone drying, which was consistent with the theory. But later on, as I progressed through the experiment, the ABA concentration in the leaves actually decreased. And the reason for this was that the sap flow from the roots in drying side, in the dry side, had substantially decreased. So the signal was no longer being transmitted to the shoot. And that's been the basis for the suggestion of alternating the wet and dry parts of the root system. And so there was the statement about there's been nothing magical about alternating partial root zone drying. I dislike magic, I would say. I would always search for scientific explanation. So again, I've done one of these meta-analysis comparing the yield response of plants exposed to alternate partial root zone drying compared to those that were exposed to fixed partial root zone drying. And again, we can see that the alternate partial root zone drying plants out-yielded those exposed to fixed partial root zone drying. And again, it leaves me with the question, well, why might be that the case? Are there differences in signaling? So another experiment with tomato, 3 groups of plants. We have well-watered plants with high soil moisture, high leaf water potential, and low xylem ABA concentration. 2 groups of partial root zone drying plants with similar soil moisture, similar leaf water potential, and then we've got very different ABA status. For the plants where an alternation event was imposed, so the wet and the dry sides were swapped over, we can see substantially more ABA in the leaf xylem sap. So alternation does seem to have an impact on ABA status of the plant. It also seems to affect another plant hormone, ethylene. This is some work from one of my visitors, Juan Gabriel Perez, who came to Lancaster and looked at the accumulation of the ethylene precursor, ACC, in the roots. And this seems to accumulate as the soil dries, but particularly when you re-water the plants, particularly with re-watering the very dry roots, you get substantial increase in ethylene evolution from the leaves. We believe this is due to increased ACC export from these roots. The more recent work from Jaime Portelas, who spent a number of years in my group, he was looking at whole plant gas exchange in trying to make the link between leaf level responses and whole plant responses. And what he found was that the well-watered plants and those exposed to fixed partial root zone drying behaved very similarly. So they had a high transpiration rate, a high canopy conductance, and a high photosynthesis, whereas the plants that were alternated with partial root zone drying showed a very similar response to drying the entire profile of the pot. Lower transpiration, lower conductance, and lower photosynthesis, indicating how important it is to alternate the wet and the dry sides of the root system to maintain this signalling. Another reason I would argue that partial root zone drying alternation is required is that it has profound impacts on the root system development. So this is an early experiment, very simple experiment, where we had partial root zone drying plants and we simply determined the root biomass in the different compartments. And after a single drying cycle, not much difference in root biomass for the partial root zone drying plants on either side of the compartment. But when the wet and the dry sides were swapped over, the previously dry side showed a substantial proliferation of root growth. And while this might not be that significant agronomically when plants are grown with abundant water and nutrient supplies in the greenhouse, this could be very important within the field to allow the roots to access water from deeper soil layers. And indeed, this is probably one of the reasons why it's possible to reduce irrigation supply with partial root zone drying and still maintain the yield of the crop. The other element that I think is important within these partial root zone drying experiments is the choice of the genotype that you're working with. So here's an experiment we did with partial root zone drying in the field with two different potato cultivars. What we found was that the Maris Piper cultivar, which is, I think, the most grown cultivar in the UK, showed a decrease in yield when it was grown with fixed partial root zone drying or whole root zone drying, but we did not see this response in the Horizon potato variety that apportioned a much greater part of its biomass to root growth lower in the soil profile. So getting the management and the genetics of the crop aligned, I think, is going to be very important in rolling out these deficit irrigation techniques. So there's been the Research Excellence Framework results released today, and one of my sort of tasks in preparing Lancaster for the REF was to actually look at the adoption of partial root zone drying around the world. And it was relatively easy to demonstrate a large number of research papers from a large number of colleagues around the world. And certainly, this map shows countries that have published research papers with partial root zone drying. And certainly, in many cases, I know the people that are involved. I did find it more challenging to compile evidence that partial root zone drying has been applied commercially. But fortunately, I was able to do this. And it's interesting that the cropping systems to which partial root zone drying has been applied are either very low-tech arable systems that have employed furrow irrigation, or alternatively very high-tech horticultural systems relying on drip irrigation. So quite a diversity in terms of the application of this technique by farmers around the world. I talked to a lot of these farmers as part of my understanding in preparation for the REF impact case study that I was preparing. And, some industries adopted the technique as a transient response, like the Californian tomato processing industry. This is before they decided, well, water scarcity is such a challenge, we're going to invest in drip irrigation. I talked to a number of vineyard owners. Often they were completely sold on the science of partial root zone drying, but often employed it as part of a wider set of measures of water-saving agriculture. And often it was, chronic scarcity of water, where rainfall was much less than historical records had stimulated them to reach out to their local university for advice in terms of applying this technique. So the most recent project that I've been involved with has actually been in West Africa. And this was part of a much wider project funded by GCRF, looking at a circular water economy. And so the question that my colleagues and I were tasked with was whether using some of our understanding of these irrigation techniques can actually help smallholder farmers. And so we used a rather different irrigation technique, alternate wetting and drying in rice, but we also used alternate furrow irrigation in tomato with our colleagues in Ghana. So we're interested in doing this. Tomato paste is incredibly important in Ghana, and yet they're not very self-sufficient. And the partial root zone drying technique seemed to match the cropping system that they were adopting, which was furrow irrigation. And we got some interesting results here where our partial root zone drying certainly substantially constrained the vegetative vigor of the plants, but didn't really have a significant impact on fruit yield, suggesting that these sort of mild water stresses were changing the biomass allocation in the plant, allowing the plants to preserve yield even though they were being supplied with less water. And I'm very grateful to my colleagues in CSIR Ghana for their collaboration over the last few years. So I've got to the end of the talk. I'm not sure how we're doing for time, Rob, but just some brief conclusions from the three parts of the talk. So certainly there is sufficient ABA in the xylem to close the stomata, but it's interesting that a lot of this ABA seems to be shoot derived and then is recirculated via the roots into the xylem, where it seems to be important in controlling stomatal behavior. Even though the shoots supply ABA, roots can also synthesize ABA themselves. And I think the balance of the shoot versus root contribution to root ABA status depends very much on the prevailing environmental conditions. When the soil is dry, that should stimulate root ABA biosynthesis. If the atmosphere is dry, that should stimulate shoot ABA biosynthesis and transport to the root system. And in the roots, ABA accumulation has a number of impacts, changing root hydraulic conductance, changing root elongation, but also seems to be involved in the process of riser sheath formation. Lastly, hopefully I've demonstrated that irrigation placement can profoundly affect plant physiology and that this can have some agronomic benefits. I think it would be naive to suggest that all of this is due to ABA signaling, but certainly root to shoot communication by plant hormones can be altered by partial root zone drying. And lastly, just a big vote of thanks to various colleagues with whom I've worked over the years and also the funding bodies. So thanks for your attention, and I hope that's been of interest.
Thank you for a very interesting talk, Ian. So first of all, I'd like to open the floor to questions. If anyone has a question, please raise your hand or alternatively type it into the chat box. Tim.
Inevitably, sorry. Hi Ian, thanks for the talk, really good. I just wondered, when you close stomata, obviously you're not just affecting the transpiration rate and the amount of water lost, you're affecting lots of things. So things like loss of heat from the leaf surface, delivery of nutrients to the shoots as well, but also carbon fixation. Have you got any thoughts about the interactions between water stress, closing stomata, and other impacts, particularly with respect to nutrients, and whether that has any implications on the way that roots behave when the stomata are closed?
Yeah, I think there's good evidence, Tim, in the literature that substantial stomatal closure has an impact on nutrient supply to the leaves. I guess this is, to look at a more sort of commercial angle here, I think this is where the partial root zone drying is potentially quite beneficial, because not only can it result in partial stomatal closure, but also substantially enhance root growth. And I didn't put the slide in today, but Fulai Lu's group in Denmark has also shown substantial impacts of particularly alternate partial root zone drying in enhancing nutrient status within the soil, but also nutrient uptake by the plant. And so consequently, I think, judicious irrigation management, you can minimize perhaps some of the negative impacts of stomatal closure. And again, I would stress it's partial stomatal closure that we are trying to enact with irrigation management, or indeed by changing the genetics of the plant. One thing that, because I've not spent enough time in the field, is just how ubiquitous this luxury transpiration is within the field. Clearly, if you're on the photosynthesis versus transpiration part of the curve that is linear, stomatal closure is almost certainly going to restrict yield. Okay, but if you're on the other part of the curve where you're getting this luxury transpiration, this is where I think there's substantial benefits to be had from partial stomatal closure. As long as you're not restricting the nutrient availability of the plant, or indeed resulting in excessively high leaf temperatures that might be deleterious to the plant.
Yeah, there's clearly some tweaking to be done to get this system optimized, right?
That's right, and that tweaking, I think, can take many forms genetically. I think the sort of tweaking that might be required from partial root zone drying may be beyond the willingness of many farmers. If there is no choice but to implement some of these depth irrigation techniques, then I think they're more happy to do that. But inevitably, the question comes up, when should we alternate the wet and the dry size within partial root zone drying? Now, I've got a pretty good physiological idea of what you should do, but actually how that translates into actual cropping systems where you've got both lateral and vertical heterogeneity of soil moisture, I think it's a lot more challenging.
Okay, so I think the next hand up was yours, Derek.
Ian, thanks for that. A lot of this is kind of not my area, but I was curious about this and some background reading. ABA just seems to be so intercalated in everything. To manipulate it seems to be a nightmare. But reading off to the side there about people speculating on the use of ABA receptor agonists to play around and manipulate crop quality. Any thoughts?
Yeah, I'm aware of quite an amount of commercial work on ABA receptor agonists currently being done. I think that Plant Impact, who you might know, they're a company based at Rothamsted. They've got some work going on the ABA receptor agonists at the moment. I haven't kept up with that literature as well as I should have. Again, I would stress though that I wonder how long-term some of these effects might be. And that's going to be absolutely critical, I think, in terms of determining whether they're actually going to be commercially viable or not. Something that is relatively short-lived that you might, for example, spray on a plant in response of a heat wave, as long as it doesn't result in excessive thermal stress, might be beneficial for the plant. I did some work with Sue Abrams a number of years, when I was still in Brisbane actually, with not ABA agonists, but molecules of ABA that had been altered by putting a couple of groups on them to make them more resistant to metabolism. And certainly I could demonstrate that with some of Sue's compounds, I could keep my plant unwatered for weeks. It just sat and looked at me. It didn't transpire at all, okay? It didn't grow, okay? So, again, my sort of take-home message there would be, well, how long-lived are some of these chemical applications going to be? A short-term tweak under acute stress conditions, if the weather forecasting is good enough, I think has some merit. The longer-term impacts, I think we need to give serious thought before we might tweak or we might, apply these compounds.
It did kind of look like a hit and run strategy.
Yeah, I mean, I think probably with a lot of and not just ABA, a lot of chemical application strategies, that there is an element of suck it and see. Often, I think there can be beneficial effects if the timing is right. In terms of timing, I would argue that there's much more precision to be gained from, say, developmental induced promoters, or indeed tissue-specific promoters. And I guess that's why I put the rootstock story in there, because I think that's an early example of a very tissue-specific process that we've manipulated.
Cheers.
Thank you. Raoul, you had your hand up, but it's gone down again now. You still want to? Yeah, go ahead.
Yeah, clear. You mentioned that there's a complicated interaction between the plants, soil management, everything. So do you anticipate it needed a specific solution for each crop and soil combination?
I'm not sure that a specific recommendation for all crops is that necessary. For soil types, I think it might be. Just in terms of crops initially, I'm convinced that one of the benefits of partial root zone drying is this change in biomass allocation within the plant. I think I provided a nice example of that with tomato at the end, where vegetative growth was restricted, but fruit yield was maintained. And I think there's good evidence from some of the Chinese groups to show similar impacts of partial root zone drying in terms of grain allocation to cereal crops. I did some work within a partial root zone drying project on lettuce, okay? And there, if you're prioritizing root growth, by necessity, you'll end up with less leaves, okay, in terms of biomass allocation. So I wouldn't be advocating partial root zone drying for salad vegetables per se. The soil type is an interesting question, and this is something that's sort of been, at the forefront of my mind for a number of years. A lot of these experiments we've done with partial root zone drying have been in the laboratory with very artificial substrates. Okay, there is evidence, I think, in the literature to show that the impacts of partial root zone drying can be attenuated or exacerbated according to the soil type. Now, part of that may be due to the distribution of moisture in the soil. Okay, so cracking clay soils, I would suggest it's very difficult to do partial root zone drying experiments because you don't get that discrete wetting and drying parts of the root system. But from a soil textural perspective, I think there's some evidence from potato crops, I think in experiments done in Peru, that partial root zone drying was just a complete disaster. You substantially stress the plant by withholding water simply because you did not get good root soil contact, okay, and good water uptake. So from that perspective, I think some of the work on rice sheath development is something that we need to give some attention to understand how important this might be, particularly within sandy soils that often tend to be used for agriculture in some parts of the world.
Thank you.
Okay, next we have a question from Carmen. Carmen, if you want to unmute and ask your question, please.
Hello, Ian.
Hi, Carmen.
Yes, I am interested in the microbes in the rhizosphere, and I would like to ask you what is your opinion about these microbes contributing to mitigate drought threats, and if you think that they can also be contributing to partial root zone drying.
I'll deal with the final part of the question first, okay? There was a paper published by one of the Chinese groups maybe 10 years or so ago, showing that partial root zone drying changed the relationship between bacterial number and soil water content. So you got more microbes with the partial root zone drying per unit of soil water content. Now, that was, I'd say fairly primitive characterization of microbial status of the soil. This was just culturable bacteria, okay? Why that actually occurred, I wouldn't care to speculate on. I think there is potentially a lot of scope to deploy more modern methods to try and understand how a technique like partial root zone drying, whether it does and how it might shape the soil microbiome. I don't think those two elements have been properly connected yet. I think most of the work on partial root zone drying and soil microbes is aimed to measure the function of the microbes. So, for example, looking at mineralization of nitrogen within the soil, and I think particularly Fuli-Lu's group. I touched only briefly upon soil microbes today because I wanted to sort of try and keep an ABA focus to the talk today. I have done some work over a number of years with my colleague Andrei in Russia, and also another Russian colleague, Guzul Kudayarova, who works on cytokinin producing rhizobacteria. And both these ACC deaminase containing rhizobacteria that break down the precursor of ethylene and cytokinin producing bacteria seem to have shown positive effects on maintaining the growth of the plant even when it's in dry soil. I think Guzul's group is quite unique in focusing on cytokinin producing bacteria. I've not seen many other reports of the use of such microbes. And I suspect the reason for that is that you might expect additional cytokinin production by microbes might actually inhibit root growth. Okay, but at least with the ACC deaminase containing rhizobacteria, I think there's a wealth of evidence from a number of groups around the world showing that they can enhance growth and yield of crops in drying soil. I think the challenge is in their application. I mentioned plant impact, I think, in relation to one of Derek's questions. So I've collaborated with Plant Impact for a number of years on a number of projects. And, always their ethos was they were going to look at chemical impacts of biostimulants. I've recently got a project with them where we're actually looking at ACC deaminase containing rhizobacteria. And, I'm pretty convinced that ACC deaminase containing rhizobacteria could have a big part to play in mitigating some of the yield losses that plants exposed to drying soil perceive. The challenge is getting them into the rhizosphere and actually stabilizing them within the rhizosphere.
In a wild microbiome.
Yeah, exactly. So, I mean, I could veer off into another talk here. In fact, I could probably give a talk just on ACC deaminase containing rhizobacteria because I've worked with them for a number of years, in conjunction with my colleagues. And, we have abundant evidence that they can enhance growth of plants in drying soil. The challenge is that once you add a single inoculant to the soil, it's got to compete with the other microbes that are there. The numbers decrease with time. Now, depending on the rate of that decrease, you may get a transient growth promotion that may be sufficient to translate into increased yield at the end of the season, okay? Particularly in short-lived crops, such as lettuce. We've demonstrated impacts or positive impacts of ACC deaminase containing rhizobacteria in lettuce crops within the UK. And we're talking like a 10-week period between transplant and harvest. So even though the bacterial numbers are decreasing with time, they're still managing to have an impact. In longer term crops, I think that's less likely unless we can rationally manipulate the microbiome to ensure that our organisms that are of interest that contain ACC deaminase are actually sustained there. I could send you a paper or two on this and I could talk all day about it, but I think it's a very relevant question.
It's really interesting. Thank you very much. I will type now literature and then wrote the reference that you gave.
Yeah, please drop me a line because we published a review on 20 years of work with one particular organism. We published it a couple of years ago. But now we're, via this collaboration with Plant Impact, we're hoping to sort of get to grips with some of the practical realities of introducing bacteria into the soil.
Very interesting. Thank you very much.
No problem. Sure.
It's great to see so much interest being generated, but thankfully there's time left for me to ask a question, which I was.
Oh, Rob.
Yeah. So I mean, obviously, when you look at from what you showed, the ABA response to the roots is very much dependent upon the root water status. That implies that there's crosstalk then from other signals. So do you have any idea about what those, what's known about what those other signals are? Presumably other hormones, anything else?
I mean, I think that, the ABA ethylene interaction seems to be quite well known based on, the work from Bob Sharp's group at the University of Missouri. But, I honestly think ABA is so important. There's probably other groups other hormone groups that it's working with as well. I didn't talk about the work today, but we've also recently got interested in ABA jasmonic acid interactions as well. And at least in some species, it seems as though that jasmonate accumulation within the roots is the primary response to water stress, and you actually need that to get ABA accumulation. I have to admit, and probably this came out with my answer to Derek as well, that I focused very much on the production of the compounds, their biosynthesis or accumulation, rather than a sort of more conventional crosstalk, what happens after perception of the compounds. But certainly I see ABA as integral with interacting with auxin, with jasmonates, with ethylene, probably cytokinins as well. So that means that any manipulation that we make in terms of root ABA status, as occurred within the tomato grafting work, often we thought the results were not primarily due to ABA itself, but ABA actually changing the status of other hormones in some cases within the shoot.
So another quick question, if I may.
Sure.
So we've recently done some work in potato on waterlogging, and we've observed ABA accumulation in the shoots there and stomatal closure. And actually, it's turned out to be much more damaging for the potato crop than drought. I mean, is that a common observation that ABA accumulates following waterlogging?
As I understand it, yes.
Okay.
And this is some of Mark Elsa's work. I mean, Mark is now at East Malling Research, but he did his PhD on root-to-shoot communication of waterlogging down at Long Ashton Research Station with Michael Jackson, and Bill Davis was his other supervisor here. And Mark's work, I think, clearly demonstrated that ABA accumulated within the leaves in response to waterlogging, which was really interesting because actually when you waterlog the soil, you don't get the oxidation to ABA occurring, right? But what actually happens is that because of your decrease in root hydraulic conductance, the plants are less able to maintain turgor, so they're synthesizing more ABA in the shoots. So it's not so much that root-to-shoot signaling is involved in waterlogging, at least for ABA, but it's this decrease in root hydraulic conductance that is resulting in turgor loss, and so you get substantial ABA accumulation. It'd be really interesting to see, having for your waterlogging experiments, whether some of that ABA, assuming that it's synthesized in the shoot, is actually being transferred to the roots and or the tubers.
Yeah, absolutely. Yeah, that's the next step, Ian.
Okay, right. I'm getting ahead of you there, Rob. But yeah, I mean, it is interesting. When I was searching the literature in preparation for this talk, I thought, well, I did want to sort of at least address the idea that you can manipulate ABA status within the plant. Okay, so I started looking at all these papers that have overexpressed NCED, and obviously I'm very familiar with Andrew's tomatoes because I've worked with them over the years. I was familiar with the rice one, and that soybean paper came up where it does seem that additional ABA is deleterious to the plant when they are waterlogged. And it sounds as though your observations suggest as much as well.
Absolutely, yeah. Okay, so we are approaching time. I'll give people one last opportunity to ask a question. If not, I'd like to thank you very much for spending best part of an hour and a half with us this afternoon, Ian. It's very interesting talk and stimulated a lot of discussion.
It's a nice break from the marking, Rob. I can which I'll return to after I close down my computer.
Enjoy your marking, and if you need a break in 2 weeks' time, I'd like to just point out to those here that in 2 weeks Christine Foyle will be speaking, and Christine will be introduced by Derek Stewart. So thank you again, Ian, and I hope the marking doesn't get you down too much.
I've got something to look forward to in 2 weeks' time, because I can get to listen to Christine. Unfortunately, I've missed the previous talks in the series, but I understand they're recorded. Is that right?
That's right, Ian. Yeah. You'll be able to find them on the APGC website.
Yes, I'll certainly be looking forward to catching up with some of the previous talks, because it seems like an excellent series you're running here, which is good.
Thank you very much. Thanks all.
Bye, everyone. Thanks for your interest.